Dear viewers, Assalamu alaikum. As I am going to discuss about the use of seq module, therefore it is important to have an idea of the classes available in this module. This module contains the following classes. As is evident from this table, for a common researcher or a student, most of these classes are useless. Therefore I will ignore them. You only need the seq class of seq module. I will also discuss about the Mutableseq class. Now let's come to discuss these two classes. First, let's see what methods are available inside these two classes. To see the methods in seq class, just type this and hit enter. You will see this list of methods. To see the methods of Mutableseq class, type this command. This list will appear. These are Mutableseq class methods, and these are methods of seq class. Most of the methods are common to both the classes. Methods in bold are only found in one of the class only. Okay, you saw methods. How to use them? Let's learn. To use these two classes, you have to follow these steps. First of all, you have to import the module using this command. Next, create an object with your sequence stored in it. For example, you stored your sequence in a variable, DNA-seq. Now you have to create another object which is specific to the class. You convert the DNA-seq object into a seq class object. You have to use this command to convert it into seq class object. You either apply this command on the DNA-seq object you just created in the above step, or you can directly use this sequence and make your class object. The last step is to apply a method to this object. For example, you want to translate the sequence. Then just type the object name, put a dot, and type the translate method. Or, you can also take this whole thing and apply the method. You can also use the method directly on this thing. Sometimes you want to save your output in some container and you don't need the immediate output. Then just type this. So your output is now saved. You can see the results anytime you want by using print command. Or, you can use the result in further sequence analysis. There are a variety of strategies you can apply a method to a sequence. Here is the list of different strategies. I will use these steps in using the methods of the class. You just saw the use of a translate method. Now let's see all the methods. You stored a sequence in an object, then you applied a class on that object. You can apply your desired class, that is, seq class, or Mutableseq class. Now type the name of the class object, put a dot, type the name of the method. This is the list of methods you can apply to your sequence object. Now let's come towards the use of each individual method on a sequence. Let's have a detailed look on each method separately. Method number 1, the complement method. What does this method do to your sequence? It creates the complementary sequence of a given sequence. That is, A is converted to T, C to G, T to A, and G to C. Before using complement method, we have to import the bio.seq module using this command. Store your sequence into a variable. In this example, we stored it in an object called input. Now convert your input sequence into a seq class object using this command. Now use the complement method. You will get this output sequence, which is complementary to your input sequence. If you store the results into a container, then you will have to use the print command to see the output. Remember, you can also use this direct method to get the same output. The direct method is a single-step method. Now let's come to the second method, that is, complement_RNA. This method is similar to the complement method, except a single difference. That is, instead of T, you will get U in the output sequence. Because the output is RNA-like, all the steps are the same. This is your input sequence. And this is your output sequence. This is the one-step direct method. Reverse_complement method. This method gives you the reverse complement of a sequence. That is, first it reverses your input sequence, then takes the complement of the reversed sequence. Here are the steps to follow to get the reverse complement. Steps are the same as used for the last methods. Here is your output. Here is the one-step direct method to get the same output for the input sequence. You can compare the output with the input sequence. Reverse_complement_RNA. It gives the same output as you got from the last method. It's similar to the last method, with one exception. That is, instead of T, you will have U in your output sequence. Let's understand it with this example. This is your input sequence. Create the class object. Apply the method on the class object, and save the output in output object. Use print command to see the output. Here is the direct method to achieve the same objective. Compare your output to your input sequence. Find method. With this method you can find the location or position of a single residue or a specific pattern of residues in a given sequence. In this sequence the first C from the left side is at position 3, as Python starts counting from 0, therefore the actual position will be 4. Suppose this is your target sequence. You want to find the location of the pattern, A, T, C, 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 G. As you can see, there are 3 such patterns in the sequence highlighted in yellow. This method finds the location of first pattern from the left side. Now create a class object. Type this command with your pattern as an option. When you print the output, it will be 8, which is 1 less than the actual position. So our actual position is 9. Let's count. So the first nucleotide of the pattern from left side is 8. Here is the direct method. If you are interested in finding the position of a nucleotide or a pattern from right side, then the method is rfind. For example, the first C from right side is at position 6, which is actually position 7. Let's take the same example as taken in the last slide. Again, there are 3 similar patterns in the sequence, but this time, rfind will locate the position of the first pattern from the right side. Your result will be 35, which is 1 less than the actual position. So actual position is 36. You can count and confirm your results. Here is the direct method. The index method works similar to find method. It finds the position of the first pattern from left side as explained in this example. Similarly, the output of rIndex is identical to rfind method. Transcribe method is used to find the RNA sequence of a given DNA sequence. In simple words, it just replaces all the T's of your DNA into U's. Here in this example, you can see that this is our input DNA. After you go through all the steps, your output will appear like this. Direct method is here. You can see it is the same sequence as above, except you have U's instead of T's. Back_transcribe is the reverse of transcribe method. This method will convert an RNA sequence into its DNA sequence. In simple words, it only replaces U's with T's. Next is the translate method. You must have heard about central dogma. It is about transcription and translation of a sequence. Transcription is the synthesis of messenger RNA, and translation is the synthesis of protein sequence from messenger RNA sequence. This translate method gives you the protein sequence as its output. It takes either RNA or DNA sequence as its input. Suppose this is our input sequence. After going through the steps, you will get this output, which is a protein sequence. Here is the direct method. This method's objective is although simple, but the mechanism it follows is a bit complicated. Therefore, I am going to give you several examples of this method so that you understand the logic behind this method clearly. The first thing it does is to remove the spaces from both the ends of a given sequence if present. Suppose this sequence has spaces on both the ends. After you apply this method, you will get a sequence with spaces removed from both the ends. Same thing has been shown in this example. Now let's come to the complicated part. In this example, I am going to show you how it removes unwanted nucleotides from the given sequence. Remember, it removes the nucleotides from both the ends, if present. Suppose this is your input sequence. You want to remove A's from both the ends. Follow these steps. At the end you will get this sequence, with A's removed from both the ends. Let me explain how it works. This is our input sequence. First, it will match A on both the ends of the given sequence. If it finds one, it will delete it. As you can see there it's an A at the left end of the sequence, so that A will be removed, but on the right side we have T, so it will not be removed. And also, the method will stop looking for A at this end anymore. You will be left with this sequence now. Now it will match A with the left end of the sequence only. As you can see, there is no A left on the left end of this sequence, therefore, it will stop further iterations on this end too. So our final product will be like this. Now let's take a different example. Again we want to remove A from both the ends. Follow these steps to remove A's from both the ends. Your end product will look like this, with A's removed from both the ends. This is the direct method. Let's explain it. This is your input sequence. First, it matches A with both the ends of the sequence. As you can see there is an A at both the ends of the sequence, therefore, both of them will be removed, and as a result, this sequence will left with us. The method doesn't stop here. It will match A with both the ends of the resultant sequence. As you can see there is no A left on both the ends, so now it stops iteration, and this is our final product. This example will further clarify your concept about this method's mechanism. Here I take a different sequence. After you apply your step-by-step commands, you will get this sequence as your output. Let's explain it again. In the first round, the A is matched at both the ends, so it is removed. In the second cycle, it finds A only on the left side. So, that will be removed. As it didn't find A on the right side, therefore further cycles on the right side will stop. In the third cycle, it will look for A on the left side only. It doesn't find any A on left side, so further iteration on left side will also stop. So this is our resultant sequence. Let's take another sequence. Follow these steps. You will get this as your result. Here is the step-by-step explanation of the cycles this method follows. After all the cycles are finished, we will left with this sequence. Now let's come to a more complicated example. In this example, I will show you the removal of two nucleotides. For example, you want to remove the terminal nucleotides from both sides if it is either A or G. You have to type the command like this. The order of A and G doesn't matter.
You can also type it as G, A. Both will work the same way.
The final strip sequence you get will be like this. Now let's understand it. This is suppose your sequence. First, it will look for A and G both at both the terminals. As you can see, both the ends have A. So, this A will be stripped from both the ends, and as a result, you will get this shortened sequence. In the second cycle, it will again look for A and G, both. Again it finds A at both the ends and remove it. Now we will get this sequence. In the third cycle, it will repeat the same process. This time, it finds G at left end and A at right end. So both of them will be removed. You will get a further shorter sequence. In the fourth cycle, again, it repeats the same thing. This time, it finds A at the right end, but there is T at the left end. So it will strip only the left A on the right side, the cycle will stop, while on the left side, it will repeat another cycle. In the next cycle it didn't find A or G at the left end. Therefore the cycle will stop at that end too. So, as a result this sequence is left. Let's take 3 nucleotides this time.
A, T, and G.
Suppose this is your sequence. You will type this order of commands.
Here the order of A, T, and G has no effect. You can type it as T, G, A, or A, G, T, or T, A, G, etc. The result you get will be like this.
Here is the step-by-step mechanism it will follow in stripping the nucleotides from both the ends. It will strip the terminal nucleotide, if it is either A, or T, or G. If it encounters C, the cycle will stop there. As you can see, at the end we are left with this shortest sequence. Because it has C at both the terminals, so no need to strip any further. Okay, now let's come to lstrip command. Lstrip works the same way as strip does, except it only strip off nucleotides from the left end only. It does not cut anything from the right side. This first example is about striping the spaces from the left side. This is the sequence of commands you have to follow. This is the direct command. And this is the resultant sequence you get. In this example, you will see how it removes nucleotides from the left side. First, let's try a single nucleotide. Here are the commands you type. This is the direct command. Here is the step-by-step mechanism. This is the second example. This is the third example. Here I use two nucleotides. Here, it will remove A and G from the left side only. These are the steps to follow. This is the single line command. Here is the explanation. At the end, you get this sequence. In this example, let's try 3 nucleotides.
A, T, and G. All the things are the same, except it only cuts these 3 nucleotides from the left side only in a step-by-step fission.
Now let's come to rstrip command. All the steps and the mechanism of action of this command is similar to the last 2 command. The only difference is that it strips nucleotides from the right terminal only. It doesn't cut nucleotides from the left side. Here is the first example. It is about the removal of spaces from the right end of the sequence. No need of further explanation. This is the second example. Here, we remove A from right terminal. Again, in this example, you remove A from the right end. This is the 3-nucleotide example. Both of these commands count the number of a specific nucleotide or a pattern of nucleotides in a given sequence. The steps to follow are the same as you followed for all the other methods. This is suppose your sequence. You created a class sec object first, as you did for all the previous methods. Now, if you want to count A in this sequence, then type this command. And print the output. The result is 8. You can see there are 8 A's in this sequence.
Now let's see if there are any A, G, in combination in the sequence.
The result is 0.
No A, G together were found.
Okay, now let's reverse the pattern.
Let's find G, A pattern in the sequence.
It finds only one.
Here is the single G, A in the sequence. Let's find the pattern T, A, T. The result is 3.
Let's confirm manually. 1, 2, and 3. Now let's try count_overlap. This does the same thing as the above command did. That is, it counts the specific pattern in a given sequence. The only difference is that it also counts overlapping patterns. But the previous command cannot count overlapping patterns. Let's try. First, let's count number of A. As you can see, the result is similar to count method. That is, 8.
Now find A, G. Again same result. Because no A, G pattern is there in the sequence. Let's count G, A. Again the result is no different. Only one G, A pattern was found. Let's count the pattern T, A, T. This time the result is different.
We got 4. Let's count. 1, 2, 3, 4. This is the same input sequence.
But this time it counts 4, because there is one additional overlapping T, A, T in the sequence.
Startswith method tells us if a sequence starts with a specific pattern. Similarly, endswith tells us about the presence of a specific pattern at the end terminal of a given sequence. This sequence starts with A and ends with U. Now, let's suppose this is our input sequence.
We want to see if the sequence starts with the pattern A, G. The result is false. Means, it doesn't start with A, G pattern. If you try A, G, the result is true.
Because it starts with this pattern. Similarly, you can use endswith method for the terminal pattern.
This sequence does not end with A, G. That's why it gives false. It ends with T, A. That's why this gives you true.
If you want to convert a sequence of lower letters to uppercase, then use upper method. The lower method does the reverse. It simply converts uppercase letters of the sequence to lower letters. Here is the step-by-step process. The join method joins two or more sequences with a spacer in between them. If these are three sequences, and this one is the spacer, then they will be joined like this. Let's suppose three input sequences. Suppose we want to join them using double asterisk as a spacer between them. First, type this command and create a class object of the spacer. Then type class object dot join and put all the three sequences to be joined. Remember, you have to put your sequences in a list form. This is the second example. We have directly put three sequences in the list. And these three sequences have been joined by the spacer. If you try a single sequence without using the square brackets, then individual letters will be spaced. Try this direct method also. Replace method replaces a specific pattern with your desired pattern in a given sequence.
For example, you can replace all A, U with G, C. Let's take another example.
The replace method requires two arguments. For example, here we want to replace all A's with K's.
You can replace double A with single K. You can replace triple C with triple G. You can replace the pattern T, A, T with G, C.
If you want to delete a specific symbol or a character or a nucleotide from the entire sequence, then use ungap method. This method's main function is to remove the gaps from the sequence. From gap, we mean the dash symbols in the sequence. So, by default it will remove all the dashes from the sequence. Here is the sequence with dashes, which means gaps. And here is the resultant ungapped sequence. Let's suppose this is our input sequence with several gaps. Create a setClass object. Then type setClass object name dot ungap and hit enter. All gaps are removed. The dash symbol is optional here. It is a default character in the method. However, if you want to remove other letters from the sequence, then you will have to specify that character in the gap method. For example, you want to remove all the A's from the input sequence. Then type this. Remember, it will remove only A's, not the dashes, so this will be the resultant output. Here is the single line command. If you have created a setClass object, and you want to convert it to Mutableseq class, then you have to use this method. For example, take this input sequence. Create a setClass object first. Now if you want a Mutableseq class object, then use this command. And your object's class is now changed. If you have created a Mutableseq class object, and you want to convert it to setClass object, then use these steps. First you create a Mutableseq class object, then you type that object name, dot, to sec, and hit enter. You are done. This and rest of the methods are only applicable to Mutableseq object. To use these methods, first you have to create a mutable class object from your sequence. Then you will be able to use these methods. Remember, you cannot use these methods on setClass object. If you want to use these methods on setClass objects, then you have to convert them to Mutableseq class object first. Then you can use these methods. This is append method. With this method you can add a single nucleotide at the 3 terminal of your sequence. For example, letter U is added at the 3 end in this sequence. To use Mutableseq methods, you have to import the Mutableseq class first. Suppose this is your input sequence. First create a Mutableseq class object using this command. Type the object name, that is, DNA.append. Inside parenthesis input the desired nucleotide you want to add at the 3 end of the sequence. Here I type K, suppose. The output sequence will contain K at the end now. Here is the direct method. By using append method you can only add a single nucleotide at the end. However, if you want to add an oligonucleotide at the 3 terminal, then use extend method. Suppose this is your sequence.
You want to add the pattern T, T, C, C, G, G at the 3 end, then use this command.
The oligonucleotide is now added. You can use this direct method also. With append and extend methods, you can only add nucleotides at the 3 end. You cannot insert a nucleotide at a specific position inside the given sequence. For this purpose, you use insert method. Insert method requires 2 arguments. The first argument shows the position where you want to insert a nucleotide, and the second argument is the nucleotide which you want to insert at that specific position. This is suppose your input sequence. You want to insert K at 5th position. Use this command. K will be inserted at the 5th position. What if you want to remove a nucleotide from your sequence? You have to use pop method. Pop method requires a single argument. You need to mention the position of the nucleotide you want to remove. Pop4 will remove nucleotide from the 4th position. Pop0 will remove the first nucleotide. Pop-1 will remove the terminal nucleotide. Pop5 will remove the nucleotide from 5th position. Actually it's 6th position, but Python call it 5th position, because it starts counting from 0. Here is the direct method. Here we remove the 3rd nucleotide from the right side. You have to type -3 for this purpose. There is another method for removing a nucleotide from a sequence. It is remove method. In pop method, you give the position of the nucleotide which you want to remove. In remove method, you give the nucleotide which you want to remove. If you want to remove G, then type G as your argument. Remember, it will only remove the first, G, from left side. Let's take another example. Create mutable class object first. Type this to remove the first A from left side. Type this to remove the first G from left side. Type this to remove the first T from the left side. Here is the direct method for removing the first C from left side. This method reverses your sequence. Means, it flips your sequence horizontally. Here are the steps to follow. You can see the output is the reverse of the input sequence. Here is the single line direct command to reverse the given sequence. So that was all for today about Seq and Mutableseq classes. Before closing, I would request you to please subscribe my channels. Thanks and bye.